the proximal convoluted tubule doing the majority of reabsorption due to the idea of sodium reabsorption, and water tends to follow that. And by the time we leave the proximal convoluted tubule, we've lost a lot of volume, but really we have not changed the concentration of the filtrate, what we call the osmolarity. Blood's normal osmolarity is 300 milliosmoles, and so is the filtrate in the proximal convoluted tubule, because it just came from the blood, so it should have the same osmolarity. As that fluid goes down the descending limb of the loop of Henle, the thin part is only permeable to water, so we're taking water out due to osmosis, because the neighboring tissues that are right next door to the loop must be a little more hypertonic than what's in the loop. And it's simple rules of osmosis. And then turn the corner, this is what the maximum amount of osmolarity you'll get, around 1200 milliosmoles. But then we go up the ascending limb, and now we change the rules. Now in the thick portion, we're no longer permeable to water because we don't have aquaporins, but we do have these symporters, these transporters to remove ions from the filtrate into the neighboring tissues, and that's what creates the osmolarity or the hypertonic environment to bring the water out. So as water goes in here, it's because of the ions that are leaving the filtrate here. And then that water will eventually end up back into your bloodstream. So as the filtrate goes up, I transport ions out. I become more and more dilute until I get to the beginning part, where will be then the distal convoluted tubule. Remember that what's really happening, though, is that we're bringing in 2 chlorides, a sodium, potassium, and the net loss is just chlorides and sodiums. The potassium just came along for the ride and got pumped right back into my filtrate. There's really no change in potassium concentration. So let's get then to the distal convoluted tubule.
Can I ask a question about
No, you can't. Not at all,
one of the
Katie.
previous ones?
What?
So in the thin part of the ascending loop, where it changes from 1200
To
to 900, is that just passive transport of the ions out then?
It's actually still a little bit of yeah, let's keep it simple. Yes.
Okay. Because that would be why there's concentration from.
Mm-hmm. Yeah, let's keep it simple and make it that. That works just fine for me. Okay. Now, one big thought. In the proximal convoluted tubule, its main job is to make phone noises. All right. Main job of the proximal is reabsorption. That's its main job. All right. Most of your 99% of your organics are reabsorbed. 108 liters of water are reabsorbed. It can do a pinch of secretion. Usually things that are secreted are like toxins in your blood or metabolites from medicine. They're just too big to fit through the filtration slits of your glomerulus. But major job of proximal is reabsorption, pinch of secretion. Now we're at the distal. We flip it. Major job is secretion with a pinch of reabsorption. Because we're down to about 20% of the volume of our filtrate to begin with. But also, this distal convoluted tubule is where we're going to begin to be under the influence of ADH and aldosterone. Up until now, these hormones will not affect the renal tubule, but now they will begin to cause a change in your ability to reabsorb. On average, we have about 9 liters of water getting reabsorbed in the distal convoluted tubule. But we can greatly change that. We can make it a lot more with these 2 hormones, the ADH and aldosterone. How can we do that? So the purple part shows us where we are, right? This is the distal convoluted tubule. Now remember, this is not anatomically accurate, because the distal convoluted tubule should be coming up between the afferent and efferent over here. This is just a schematic functional diagram, so you can see what happens in various parts of the nephron. And you got a split screen. And you always want to pay attention to things, be really nitpicky. And as I look at this, I realize there's only a few little purple bumps here, which represent microvilli. Compare that to look at all the microvilli, right? And that tells me right there, that's why it doesn't do a whole lot of reabsorbing. It has a much lower surface area than the proximal convoluted tubule does. Now, get your bearings. That's the lumen, that's the apical side, that's the basal side, peritubular capillary. And what do we see? We see potassium leak channels and a chloride sodium symporter, passive transport, no ATP. It simply works by diffusion, right? Because the pump is what maintains your ability for passive transport. This does require ATP. Keep in mind, this is your friend that you know so well now, your 3-sodium 2-potassium ATPase pump. It requires 1 ATP. They're just being lazy and not showing you the 3 sodiums, 2 potassiums. Here's the nice rule. All ATP pumps are always on the basal side. Channels are always on the apical side for sure. We can also get some on the basal side, but there are no pumps on the apical side. And here's how it works. If sodium comes into the cell becomes more concentrated with sodium. To maintain that ability, I pump the sodium out. Well, if you remove sodium from the filtrate, does the filtrate become more hypo or more hypertonic? More hypo. The cell becomes more hyper, right? And that would influence water to come into that cell. And that pumped the sodium out, and what they're not showing you is that water would also follow the sodium. That's the rule you want for kidney function. Right? Sodium and water. It's like a puppy following a kid, and the puppy is the water and the kid is sodium. Water always follows sodium. Now, if I give you a hormone called aldosterone, right? Look what it says up here. Sodium-potassium exchange in aldosterone-sensitive portions of the distal convoluted tubule and collecting duct. When you have aldosterone present, you increase the number of sodium-potassium pumps in the membrane. What they really shouldn't be showing you is that that's not a pump. What you want to put in there are more sodium leaky channels and more potassium leaky channels. You want more sodium and potassium leak channels in the apical side, and you want more pumps on the basal side. And the more aldosterone, the more of those you put in. And the more of those you have, the more you can reabsorb sodium. That will end up in your blood. But there's a cost in doing this. This is what's known as the sodium-potassium exchange. As I am reabsorbing sodium under normal conditions, I exchange it for potassium ion. So if I have aldosterone, I have more sodium leak channels and more potassium leak channels, I can get more sodium to diffuse in. I've got more pumps to pump that more sodium out, but those more pumps will also pump in more potassium, and I have more leak channels for potassium to go back into my filtrate. You just do more of everything because you have more channels. Does that make sense? All right, you want to anchor to that. Think about that for a moment. That's going to be a big key idea. Under more aldosterone, you get more sodium reabsorption because of more leaky channels and more pumps. You also get more potassium secretion because you have more leaky channels for potassium and more pumps for the sodium. So you get a sodium-potassium exchange from filtrate to blood and blood to filtrate. Now, in terms of the secretion part of the distal convoluted tubule, we just mentioned potassium. We're also now going to look at sodium secretion because of sodium reabsorption. Let's see how this works. So we're still in the distal convoluted tubule, right? Always read headings and get your bearing, like what are we looking at, right? Hydrogen secretion and bicarbonate reabsorption along the entire DCT and collecting duct. We're going to kind of simplify this a little bit. Let's just first look right here. There is a sodium-hydrogen exchanger. Just like a sodium-calcium exchanger for the heart, right? This is passive, but it is still taking sodium out of the filtrate and putting it into the cell. What they're not showing you is your normal, bless you friend, sodium potassium ATPase pump. It's there. So if you need to draw one in right now, do it right now so you don't forget. It's there. That's how I can get rid of that extra sodium coming in due to the sodium hydrogen exchanger. So if I have an exchanger that takes sodium out of the filtrate into the cell, and then my imaginary pump to pump the sodium out, the more sodium that can come in means that the exchanger can work faster and I can get rid of more hydrogens. Well, I have another exchanger down here, another sodium hydrogen exchanger. So as more sodium leaves the filtrate and goes into the blood, it costs me more hydrogen ions from the blood into the filtrate. And that means you're changing the pH of blood. It means you're changing the pH of the filtrate. So the more sodium you reabsorb, the more alkaline the blood becomes, and the more acidic the filtrate becomes. We're not going to worry about the ammonium chloride pathway. We've got enough to worry about as it is, right? So we're not going to worry about any of this over here. All right? We will see this when we get into the digestive system, and a little bit later in tonight's notes, but we're not going to worry about this actual diagram here. Now I want to go back to this idea. Right? Sodium hydrogen exchange. Why does aldosterone stimulate hydrogen ion secretion? When we go back to what aldosterone does, I already mentioned this to the first lab group. If I asked you on page 3 of your quiz, what is the function of aldosterone? You're going to tell me that it puts more sodium and leak channels, more sodium potassium leak channels in the apical membrane and more pumps in the basal. That's what it does. That's its function. Right? More sodium and potassium leak channels in the apical membrane and more pumps in the basal. That's what it does. Now, look what happens if you do that. If I have more sodium leak channels, can you see how you get more sodium to leak into the cell? All right, we got to do this one step at a time. So you guys all see that if I have more sodium leak channels, I can get more sodium to diffuse into the cell. All right, now, because of that, this exchanger can work faster. Remember the rule. When chemicals bind to proteins, proteins change their shape. If I am this sodium hydrogen exchanger, right, I'm going to be kind of doing this idea. Right, so here's hydrogen, here's sodium. What caused me to change my shape is that I had to have both things attached to me. If I have hydrogen attached, I'm going to wait for sodium. And until sodium lands on me, I won't change my shape. So if I don't have to wait very long, I can do this a lot faster. Then I go back to my, original shape. Right? So I'm exchanging, right, hydrogen for sodium. I go back to my original shape, wait again, go back to my original shape. So if I have to wait a long time for sodium, I'm going to be here a long time. But by having more sodium leak channels, that pump or that transporter, this transporter, doesn't have to wait very long. Which means I can actually get more hydrogens into the cell. Which means more hydrogens leave the blood and go into the filtrate. Which means the blood becomes more alkaline and the filtrate becomes more acidic. All because of aldosterone simply putting in more sodium leak channels. More sodium means that guy can work faster.
How does that work? Are the channels already there and aldosterone just stimulates them to open, or how is this more complicated?
In a way, if you have the McKinley book, he actually, and I don't know why I didn't put this, it's kind of my fault, I don't know why I didn't put this in your notes, but aldosterone is a steroidal-based hormone. And you go, okay, who flipping cares? Steroidal hormones, they don't bind to membrane-bound receptors, right? They go through the cell and attach to an intracellular receptor, which goes to the DNA to activate transcription of proteins, and you build more channels, and then you transport those channels with exocytosis, and you plant them into the membrane. That's how it works, right? she asked.
So, for the purpose of that question, why does aldosterone stimulate the hydrogen ion
Yeah.
secretion? It's because the sodium is landing on that pump, making it go.
Well, because you put in more sodium channels, you increase the rate of sodium diffusion into the cell. If more sodium diffuses into the cell, this transporter can work faster. If that can work faster, then I have more hydrogen loss along with more sodium reabsorption.
Yes.
Okay.
When you say more hydrogen loss from the blood going back into the filter.
Mm-hmm.
Okay.
Yep, that's what's known as secretion. Remember, secretion is from blood into the filtrate. The reabsorption is filtrate into the blood.
Okay.
All right. Remember we did it in the endocrine section, and we talked about how PTH and calcitonin influences the kidney's ability to put calcium in the urine. And I never really did tell you what part of the kidney actually does it. Well, now you know what part of the kidney plays a role in calcium reabsorption. It's the distal convoluted tubule. So from now on, don't say that PTH and calcitonin influence the kidney's ability to reabsorb. You're going to say it influences the distal convoluted tubule's ability to reabsorb calcium ions. A little more level of specificity. Collecting ducts can do pretty much what the DCT can do. So the DCT, the collecting tubule, and the collecting duct, right? The DCT, the collecting tubule, and the collecting duct all respond to aldosterone and ADH. They all do the same thing when they get that hormone signal. It means I can also have more potassium secretion, more hydrogen ion secretion in those regions, and they're all affected by these two we've mentioned so far. We haven't gotten to that one yet, but we will tonight, and that's also affected in the distal convoluted tubule. On the average, we get about 17 liters a day of water out of the out of the collecting duct. Not the papillary duct, the collecting duct. So you gotta really anchor to your anatomy of, once I leave the distal convoluted tubule, I go into a little teeny short collecting tubule. I go into the big collecting duct drain, and when several collecting ducts join together, we have what structure? A papillary duct, which then, remember, empties into the minor calyx. Once you are in the papillary duct, you can no longer touch the recipe. You can no longer modify urine. You can no longer change the composition of filtrate. Whatever it is, it is. We can no longer modify it. So that means we get some regional functions. We're mainly thinking about, remember, how the kidneys play a role in maintaining blood volume and then maintaining ion concentrations, right, and removal of waste. So we can divide the function of the nephron into two thoughts in terms of water reabsorption. From the proximal convoluted tubule all the way down the descending limb, right, that's water reabsorption. That's called obligatory because it's obliged to follow the rules of osmosis. No hormones for you guys. No hormones influence the blue region. It's simply due to ion concentrations. The purple region, though, is under the influence of hormones. That's why it's called facultative, because I can facilitate the reabsorption of water. Wherever you have purple, that's where ADH and aldosterone will have their function. Wherever you have purple, that's where ADH and aldosterone have their function. So don't memorize this, right? But it's a big idea of what we just saw, but it gives you a visual of increasing concentrations. So everybody just watch me for a second. So there's my 300 milliosmoles. The dots represent concentration. I know that water will leave the descending limb because of aquaporins and due to osmosis. And as water leaves, my concentration of dots increases. However, now up to the ascending, I have the ability to be permeable to ions, but not to water. So as I remove the ions, I get fewer and fewer dots. I'm now up to about 100 milliosmoles. And see, there's a few more dots here than there is there. And that concentration, if there's no hormone production, will maintain itself all the way through the collecting duct, the papillary duct, and you produce large amounts of dilute, clear-colored urine. If, however, we need to increase the concentration of your urine because we need to save water, there's my obligatory water reabsorption. Then now we're seeing the effects of facultative water reabsorption. ADH allows us to reabsorb more water. And here's what you're going to tell me the function of ADH is. ADH causes the insertion of more aquaporins into the cell membrane. That's its job, to put more aquaporins into the membrane. The benefit of that is you get to reabsorb more water. So do not tell me that the function of ADH is to reabsorb more water. Let's get more specific. Let's get down to the real reason. And that's because it puts more aquaporins in the membrane, which allows you to reabsorb more water. You want to kind of tie that idea in. This we're going to skip, right? I never teach it anyways, but I don't want to spend time going over it. But in case you wonder what is known as the counter current mechanism, that's what this refers to. It's simply the idea of this, and just look at this and get the big idea, but don't have to follow the whole thing. So there's your afferent arteriole, right? There's filtration. There's my peritubular capillaries. Here's my descending limb, and I know that the concentration gets greater as I go down because the neighboring tissues, right, are slightly more hypertonic. Well, then how come this tissue just doesn't become flooded with water? Because as the water leaves the descending limb, going to the more hypertonic tissues, the peritubular capillary is right next door. And the blood there is a little more hypertonic than the tissues are. And as long as you remember, I'm trying to pound into you guys about why things go anywhere, right? From regions of higher to regions of lower. The concentration of water here must be higher than the concentration of water there. But the concentration of water here must be just a little bit higher than the concentration there. We're going from, relatively speaking, hypo to hyper, relatively speaking, hypo to hyper. And that maintains the water to go from filtrate to tissues into the blood. And we have the same thing happening with the ions on the other side. I'm transporting ions out of the filtrate into the tissues. But they can diffuse into the blood to make the blood hypertonic, so I can draw water into the blood. It's a self-mechanism, right? It just keeps on going because we're just simply going from regions of higher to regions of lower with both ions and water. And this is the idea of how the peritubulars are wrapped around the collecting tubules. Do not memorize all the steps. Read it if you want to have it make sense, but I don't want to spend any more time on the countercurrent mechanism. You have no study guide question about it, and you will have no exam question about the countercurrent system. All right, that's where I wanted to have ended Monday night. Now, we need to look at how we can maintain a normal glomerular filtration rate, which I'm sure all of you can quote to me as 125 milliliters a minute for 180 liters per day, right? Right? Good, good. Now, how can we keep that the same? There are three major mechanisms your kidneys will use to make sure you keep your GFR at 125 mils a minute. I can do either autoregulation, a hormonal response, or the autonomic nervous system. Those are my three choices. The first one is autoregulation. Responds to local changes in blood pressure and osmolarity. Triggers changes in the diameter of the afferent and efferent and diameter of the glomerulus. It's a reflex response. No hormones, no neurotransmitters. Muscles simply contract and relax. I'm going to walk you through how that works. The autoregulation is a second-to-second, minute-to-minute response of your kidneys. So if I were to have Michael stand up and run up and down the stairs several times, his blood pressure is going to change. If your blood pressure changes, so does your GFR. You don't want it to change. So autoregulation kicks in and says, oh, I can fix this, and I'll fix it. But if for some reason it won't fix it, after about a day or two of inappropriate responses, we go to the next level of effort. That's the hormonal pathway. So round 1 is autoregulation. That doesn't work to go to round 2, which are hormones. We're going to walk you through how each of these are going to work. So let's look at first the bottom one, the autonomic nervous system, just the sympathetic, because there's an argument as to whether or not there are actually parasympathetic fibers in the kidney. We know they're sympathetic, but some say there are no parasympathetics. And it's quite straightforward, right? What you want to do is decrease the glomerular filtration rate through the sympathetic nervous system. And how do we do that? Well, sympathetic stimulation of the kidneys leads to vasoconstriction of the afferent arteriole and of the efferent. If we have a more constricted arteriole, less blood can come in, that lowers the pressure, and that lowers GFR. We're not going to worry about this side over here. We're only going to be doing the right side, anatomical right side, dealing with vasoconstriction of the afferent and efferent arteriole. The result is decreased blood flow into glomerulus. That means decreased GFR, and we have now conserved our blood volume. This tends to be a severe response. So if you had a really severe injury, lots of blood loss, this will kick in. This does not happen on a day-to-day basis. This does not happen when you're feeling really, really thirsty. This would happen when you have been at severe thirst for, let's say, 2.5 days, and you've been sweating your butt off, and you have lost a lot of water through sweat. This is the last stage. This is like a Hail Mary pass to try and keep you from losing too much more blood volume. We, however, talk about the normal everyday things. So look at the very top of the slide, right, where it says maintaining normal GFR response to decreases in blood pressure. What happens to GFR if we have a decrease in blood pressure? It goes down, right? So that's the problem. What we want to do is we want to bring it back up again, right? You want to keep it at 125 mils a minute. It drops to 120, autoregulation kicks in and does the following idea. It's called a myogenic response. The walls of the arteriole have baroreceptors in them. And if they sense a drop in pressure, they dilate. That causes more blood to come into the glomerulus. If more blood comes into the glomerulus, what happens to GFR? It goes up, right? Problem fixed. Right? It's a reflex response, myogenic, muscle-produced response. Now, if that doesn't work, they can actually add one more level of response. They can also constrict the efferent arteriole. So I created a mnemonic. This is not in a book. This is my own weird warped sense of mind here. An autoregulation in response to a drop in blood pressure, I think of as DACE. Dilate afferent, constrict efferent. That will help me raise my GFR, and I have fixed my problem. It's simply a change in the diameter in response to blood pressure. If that doesn't work, after a day or so of trying to dilate the afferent and bring in more blood so I can increase my GFR, the hormonal system kicks in and says, all right, you had your chance. Now it's my turn. And we're going to do what's known as the renin-angiotensin pathway to cause an increase in GFR. So how do we do that? Well, here's first the idea of autoregulation. Right? Autoregulation responds to a decrease in blood pressure. What happens to GFR? It goes down, right? We want to fix that. So what do? Here's normal, right? There's your blood coming in. All we have to do is dilate the afferent. That brings in more blood volume. That itself increases the GFR. And if it didn't work, we simply just do a little constriction of the efferent, make it harder for the blood to leave. That also helps increase GFR, and we have solved our problem. It goes back up. This is a minute-to-minute, moment-to-moment, automatic response. That's why it's called auto-regulation. The kidneys are regulating their own flow of blood to regulate their GFR. Now, when that doesn't work, after about a day or two, right, we're going to do this. Don't let it overwhelm you. It's actually quite simple once you've worked your way through this. Let's go way over here where we have our little teeny liver, which you'll learn all about in the digestive system. It's producing a chemical substance called angiotensinogen. It's always making it. It's always going into the blood. Here's another little hint for physiology. Sort of like when chemicals bind to proteins idea. Any compound that ends in ogen tends to be an inactive form of that substance. We call a precursor. So this substance called angiotensinogen is an inactive chemical signal. I need to activate it. So the liver constantly produces it. Now here's the kidney, and there's your juxtaglomerular apparatus. That's your JGA that we mentioned in the lab on Monday night. It's right there. It senses the drop in blood pressure. And in response to the drop in blood pressure, the JGA releases a chemical signal called renin. Renin is an enzyme. What do enzymes do? They help to do either anabolic or catabolic reactions. It's going to help you break down angiotensinogen into its next chemical substance. But what is the name of the next chemical substance that angiotensinogen gets turned into? It's angiotensinogen, or angiotensin 1, excuse me. Angiotensin 1. So the liver keeps making angiotensinogen. In response to a drop in blood pressure, the JGA releases renin. Renin reacts to the angiotensinogen and turns into angiotensin 1. But it's still not active. It needs to be changed one more time. So, as that inactive chemical signal travels through the blood supply, right? Renin converts angiotensinogen into angiotensin 1. In the lungs and in your capillaries, in the endothelium, they release an enzyme called ACE. And ACE is, they didn't put it up there, ACE is this.